so now we're going to turn our attention to gastric secretions. Yesterday was kind of good, right? We talked about the esophagus, we talked about esophageal motility, salivary secretions, but now, as you saw from Dr. Shuttleworth starting to talk about gastric motility, things are starting to get a little bit more interesting, because now we've surpassed that element of just the simple tube to get food from the oral cavity down to the stomach. Now we have propulsion, we have mechanical action of the stomach. That's the mechanical breakdown, that grinding material, that grinding action that Dr. Shuttleworth mentioned. Now, what I'm going to talk about are the secretions of the stomach. Secretions primarily being, as Dr. Lin mentioned, acid. We're going to talk about that mechanism. And then we're going to talk about the protease that is released to begin our digestion of proteins in our diet. So now we're going to be talking about disruptive types of activity in the stomach with acid, and then chemical digestion in the stomach. So this picture is just to remind you of what Dr. McGuire talked about in his histology about the structure of the gastric wall. So the lumen of the stomach is on the top part of this picture. We have a series of cells that line the lumen of the stomach. These are lots and lots of mucus cells. We'll take a look at those in a few minutes. And then we have these very, very, very deep pits. And if you ever take a histological section of the stomach, very, very obvious where this section comes from in the stomach, because you'll see these hugely deep pits that go way down into the wall of the stomach. Now, why? Why do you have gastric pits? Why do you have these cell structures so far down and deep away from the lumen of the stomach? What's the reason you can think of? Don't make me call on somebody. That was really good, wasn't it? You know, I have to admit, I proposed doing that several years ago, like in law school, and our associate dean, Dr. McGuire, he's not here today, he said, unless you want to be tarred and feathered, I would not recommend it. Okay, so I won't call any individuals out. Sometimes I might stare at people awkwardly, waiting for a response. So why do you think, generally, why do you think those pits are so down deep?
Keep them from the acid.
Yeah, exactly. Keep the cells protected from the lining of the stomach. Now, the lining of the stomach, we're going to talk about these particular cells, the mucus cells, because those are strictly involved in protection of the stomach wall, the gastric wall. Everything in the gastric pits is the business end of the stomach. So these are the particular cells that we're going to talk about, the parietals, the G cells, ECL, and chief cells. Those are the primary secretory cells that are going to be producing the factors that are going to be involved in digestion. We put them down here in the gastric pit. We put them down there. Whoever designed us, depending on your affiliation, put these cells down in the gastric pit because these are very sensitive columnar epithelial cells, and there's very little protection down here against that harsh environment. So you're protecting against the acid, you're protecting against chemical digestion that is going on in the stomach lumen, and you're protecting against that mechanical damage that can occur. Now, how do we get this material that is secreted way deep into the gastric pit? How do we get that into the lumen? Think Dr. Shuttleworth. Motility. Motility. So it's just that constant motility of the stomach that is squeezing and pushing and pushing and squeezing that is ejecting that material out of deep into the pits and putting it into the lumen. The rugae folds inside the stomach. Why is that such a good thing for us? Think about the November holiday. The stretching, right. So November, Thanksgiving, we eat way, way more than our stomachs possibly should, and that allows us that huge expansion to be able to enjoy that meal because you've got lots of room to spread this food out. How about mechanical digestion occurring? How's that working? Not so well, right? Tryptophan, sleep it off. Yeah, tryptophan is the precursor for what? Melatonin and serotonin? Yeah. Yeah, I sleep a lot after those meals. Okay, that's a side note. I like to throw out little tidbits that will impress your friends on Friday nights kind of thing. Okay, so we're going to talk about 4 primary secretions of the stomach, and these are the major 4. Now there are others. There are some hormones in there. Those are lesser known. Those are known very well by research scientists, but they don't have a lot of clinical impact, so we tend to leave those out. We just focus on the ones primarily that you have to know. First one we're going to talk about is hydrochloric acid. HCl. Second one, pepsinogen, which is going to be the precursor to our digestive protease. Mucus, to protect the stomach. And intrinsic factor, which is a factor required for vitamin B12 absorption. So if we look at some experimental data and look at acid output, what we see is that under basal conditions, before we eat a meal, there's very little acid being produced and secreted into the lumen of the stomach. So then, upon a mealtime, at this point, if you measure the acid output into the stomach, you see it rises over the course of about an hour and a half and then slowly decreases. So what this is indicating is that there must be some sort of signal and a trigger to start that gastric acid production. And then as the material, as Dr. Lynn showed and Dr. Shuttleworth showed, with evacuation of the gastric lumen, the acid goes down when food decreases. So this is an indication that it must be the meal itself that is triggering the acid release. So what is acid? I might get a little excited about this because I just think this is totally cool. I actually have asked some of my undergraduate classes, isn't it weird that we produce hydrochloric acid? How do we get that? And one person said, well, we drink it. In the future, I will keep all your business cards so you'll have clientele as people somehow think they need to drink acid. We produce enormous amounts of acid. So under basal resting conditions when we're not eating, the pH of our stomach is anywhere from 6 to 7. Now, if we remember our general chemistry, we will produce enough hydrochloric acid to drop this pH in our stomach during mealtime to 2. Now, what's important about this information? Knowing pH 6 to 7, neutrality, close to neutrality, best resting, and then down to pH 2. But remember that this is the log units. This is a log order change. So this is a gigantic, enormous amount of, literally, it's hydrochloric acid being produced. So how is the hydrochloric acid being produced? I promised I would draw a little bit bigger on my cells. So the cell type we're talking about is one of those cells in the gastric pit. And this is the parietal cell. The parietal cell is the main cell that produces hydrochloric acid. In the old literature, it's called the oxyntic cell. Nobody uses that terminology anymore, but if you happen to look in some of the older GI physiology books, they might describe oxyntic cell. That's the same thing as a parietal cell. So the parietal cell is going to be making and secreting the acid. How does that happen? You know the answer. Okay. Row 7. No. I love doing that. I have to admit, I'm sitting over here. You know, I know he's not going to call on me. And then so I glance over at you, and there's the looks of terror. It's like, no, don't call on me. So we're going to perform a chemical reaction. We're going to take carbon dioxide. We're going to combine this with water, And we are going to make this. Remember that reaction? We looked at it in the salivary gland. And the salivary gland, the purpose of this was to make the bicarbonate ion. In this case, what we're going to do is we're going to be making the proton. Of course, the enzyme that does this is our favorite carbonic anhydrase. Another tidbit for Friday night group study. Carbonic anhydrase is one of the fastest enzymes known. It has a turnover rate of upwards of 10 to the 8th per second. Wow, that's pretty fast, huh? So there's no depletion of this enzyme here. As long as we're supplying it with substrate, it's going to be making its products. Remember your biochemistry. Carbon dioxide, where does that come from? Don't say breathing. Cellular metabolism, right? Specifically, where does the CO2 come from? Wow, this is going to be a long block. I'm actually doing this for a reason, because it's helping me gauge in a couple of weeks when we start the metabolism to kind of see where you are in the landscape here and kind of where I need to make some adjustments. So the CO2 is coming from decarboxylation, oh God, reactions from the TCA cycle. So when we breathe, and the CV palm renal block, Dr. Resta was, he did the block chair for you guys, right, Dr. Resta? Yeah. So when he was talking about oxygenation, you know, hemoglobin delivers oxygen, and then your red blood cells take CO2, bring it out to your lungs for expiration for gas exchange. This is where all that CO2 is coming from. It's coming from your TCA cycle, okay? So we think about that as providing CO2 dissolved in the red blood cell as bicarbonate as for expiration, but that CO2 is also serving a lot of other vital functions, and we'll see a whole bunch of those. So this is coming from normal cellular respiration. Now, we have a proton. The proton is the one that's responsible for dropping the pH. So we need to take this proton and we need to put it into the lumen of the stomach. Now, this is an extremely important process. And so that means it's going to require a transporter because protons don't naturally go through that bilipid membrane, so we're going to have a transporter. It's a really important process. So do you want this to happen just by facilitated diffusion so that as soon as you make the HCl or the hydrogen ion, it just goes out? Or do you want to pump this thing out? You want to pump this out because of our graph we just looked at. When you have a meal, you want to acidify the stomach environment very quickly. So what we're going to do is we are going to couple the transport of this proton with potassium. Now, do you have a lot of potassium in the lumen of your stomach to drive this process? Yeah, maybe if you eat a banana. A lot of potassium in banana. So, you know, if anybody ever gets cramps, yeah, eat a banana if you're lacking potassium. So, we're going to want to pump this. So this is an ATPase. So as we looked at the transporters yesterday, this is a primary active transporter. Now, since we do have not that much of potassium in the lumen of our stomach, we could easily deplete the potassium in the lumen, and this transporter will stop. Because this transporter requires the binding of a proton and potassium in order for it to flip and switch those ions back and forth. So in order to prevent the depletion of potassium, we are going to have a potassium channel, which is facilitated diffusion. Now remember, facilitated diffusion runs on a gradient. So we are establishing a gradient of potassium, high concentration inside, low concentration out, and then that will facilitate the potassium exiting the parietal cell back into the lumen so that we can keep this potassium proton ATPase pump going. Now, another very important chemical and medical principle is that nature hates a charge difference. So if we keep pumping protons out into the lumen, we are going to have a charge differential across that membrane. So the only way that we can have charge differential in our body is because of active transport activity that maintains polarization of membranes. And those only occur in certain situations and certain cell types that control that. Everything else in the body is going to be trying to achieve electrical neutrality. Now this is important because we have to balance the ions and by their charge. This is going to become really important when we start talking about metabolic acidosis in the blood and how that works. But in this case, we have a lot of positive charge. So that means that we're going to have something that is going to drag a negative charge to follow this to neutralize that charge. And you know what this is. It's the chloride ion. And this is why in the lumen we are producing hydrochloric acid. Now we don't have a lot of chloride ion available inside the cell, so we have to get it from somewhere. So what we're going to do is we are going to put another transporter. Now this transporter will be on the basal surface of the parietal cell. This transporter will bring in chloride from the blood. Notice how I drew it. It's a dual transporter. This is also like the proton potassium transporter. It's an antiporter. And here, in order to make sure that our chemistry is balanced, we are going to be transporting out our bicarb ion. So by having these 4 particular transporters on the cell in their proper orientation of apical and basal location, we now have balanced our electrical charge. We have a mechanism of actively pumping protons into the lumen, and we have a way to get one of our waste products from our carbonic anhydrase out into the blood so that we can transport this to the lung. Now, yesterday I said don't worry about the transporters in the salivary gland. You don't have to worry about those because that's complicated to balance. Now, what's important about this? What's really important here is this potassium proton ATPase pump. This is going to be very, very important when Dr. Shuttleworth talks about how proton pump inhibitors work. He's going to talk about the chemistry of the PPIs and how it affects that pump. So how this pump works, yeah, you got to know that. The other thing you have to know is on the basal surface, because occasionally there'll be this term called an alkaline tide. And this is specific for gastric epithelium. And what this refers to is that if you measure the blood coming out of the gastric vasculature, that blood will have a slightly elevated pH. Now, we're not talking about whole pH units, but it's slightly elevated because of the extra bicarbonate ion that's being put into here in order to get the acid production in the lumen. And that is termed alkaline tide. I don't recall ever seeing any abnormal medical condition with this, but it is referred to, it's a medical terminology, alkaline tide, so it may show up. It will not show up on one of our block exams because it's just there. But just keep a little asterisk next to it for next, like March or something like that. You might have to pay close attention to March. Something's happening, they told me. Everybody knows what's happening in March, right? Okay, so now that we have the mechanism here of how acid is produced, obviously we have some sort of regulation mechanism of how to control this. And so we're going to turn our attention to the next type of cell. And I'll turn the page on this one. In this type of cell, a very cartoon character out of that. So this is going to be regulation of acid secretion. So what is controlling the activation of the parietal cell? So now what we're going to be looking at here is the parietal cell. I've drawn this squiggly little top corresponding to the image that you have in your notes, because the parietal cell, remember, is a secretory epithelial cell, which means it has a microvilli structure, often called a brush border. And this, of course, is facing the gastric pit, which is continuous with the gastric lumen. So the parietal cell's making these protons by our reaction. So what controls this? Well, the first thing we have to talk about is what we started yesterday called the cephalic phase of digestion. When you see, smell food, even taste food, your higher ordered centers, CNS, are signaling through the vagus nerve, and it's going to prime the pump. So in the salivary gland, we saw that the parasympathetic activated acetylcholine, which triggered the M3 receptors on the salivary gland to secrete their products. We are going to have the same activity on this parietal cell. So this same vagus signal will trigger acetylcholine release on M3 receptors, and we will go through our phospholipase C, IP3, increased calcium signal, and this is going to trigger the activation of gastrin activity to make protons and to release protons into the gastric lumen. So what is this doing in the cephalic phase? You have no food yet in your stomach. You've smelled it, you've looked at it, you're salivating, you're just getting ready to swallow. This is, I like to call this as priming the pump. So many of you have had to operate hand gasoline lawnmowers, and before you start it, you've got to hit the prime button there. You're putting some gasoline into the engine to get this whole thing started. That's exactly what this is. The cephalic phase is triggering through the vagus nerve to increase the amount of calcium in these cells to start acid production. Now calcium, what's that got to do with protons? Well, if you look at an electron micrograph, which will be on, I believe, on the next page, yeah, next page. What that is showing is a large nucleus in the center. There are a whole bunch bunch of little round objects that are all very darkly stained on that electron micrograph. What are those structures? Mitochondria. Mitochondria, right. Why would this cell have such a large number of mitochondria? What does mitochondria generate?
Energy.
Energy. Where is energy really important in this cell? ATPase, the potassium proton ATPase. So this cell is going to have a very, very high metabolic capacity in order to generate a lot of ATP so that pump is not inhibited. You want to keep that pump working. Now, in that picture, you'll also see a lot of little vesicles. This picture is fairly old, and they call it tubulovesicular structures. They're little vesicles. And they are located all over the cell. They have a concentration underneath the apical surface. What the calcium is going to do is it's going to cause these vesicles to translocate to the cell surface on the apical side and fuse with that membrane. These vesicles contain the potassium proton ATPase. So what the parietal cell has done is it has undergone transcription and translation of this gene to synthesize the protein and pre-package it in these little vesicles. Because remember, protein synthesis from transcription of a gene all the way down can take anywhere from 14 to 16 hours. So let's pre-package these proteins so that all we have to do is trigger through this vagal response and cause the fusion of these vesicles with the surface. And now these transporters are exposed on the surface of the cell and they become active. And once they're on the surface of the cell, now we can transport our protons. Yes?
Sorry, when you say proteins, what are you referring to?
So the transporter itself, this particular antiporter, which is the potassium proton ATPase, is the transmembrane antiporter. And this, under resting conditions, is located in these vesicle membranes. Okay? So the vesicle membrane, remember orientation, we're going to have the inside of the vesicle is going to be like the outside of the cell. And so the orientation of this would be pumping protons in, pumping potassium out. So when these vesicles fuse with this membrane, the orientation of this protein transporter is now in the correct orientation to push the protons out into the lumen. Okay? So now this allows for a very active signal. So as soon as that vagus fires, essentially you're getting acid production literally within seconds to minutes because the the pump is already made and inserted into the membrane. Now, downregulation, we have to be able to stop proton activity, secretion. We saw in our graph, after our meal empties the stomach, we see this decline. That is because these vesicles are now retracted off the surface. And they're retracted back to these vesicle forms, ready for the next meal. And so by doing this, we have a very, very rapid mobilization and retraction so we can control acid production and secretion down to the minutes. Now, what causes the retraction? What signal? Well, we'll take a look at the major signal there, but it's also loss of any positive input. We don't have a positive input. We don't have elevated calcium levels. We don't have vesicular fusion. Once our calcium levels start to drop, the fusion stops, and retraction is the automatic constitutive method to pull those back in. Okay, so that's the cephalic phase. That requires the vagus. Now the next phase is the gastric phase. This is going to require food actually entering the lumen, the gastric lumen. And that is going to signal the parietal cells to now really ramp up the acid secretion. Now we have this stepwise reaction for the simple fact that somebody could tease you with some food so that your cephalic phase triggers, but then they take that burrito away from you. And you're depressed. But you only have a little acid in your stomach. You don't have the full-blown acid production. So what will trigger that? Now we have to talk about the hormones. We have two hormones. One is called gastrin. The other is called histamine. The gastrin has a specific receptor. It's located on the basal surface of the parietal cell. Once the gastrin binds, it is going to mimic the activity of the M3 receptor. So it is going to go through the PLC activation, IP3 calcium release. So this will tie right into this signal. Now what we have is synergy, the principle of synergy. We'll talk about that in a couple of minutes. But what we're doing is we've got one signal that triggers a pathway. We've got another signal that is not triggering a separate pathway, but it's feeding into the same pathway. So picture two hoses connected to two outlets. One hose feeds into another one. This second hose feeds into that Y structure, that joint. What's the water going to be like coming out of the conjunction of those two hoses? Much greater, right? So this is the principle of synergy, and also called potentiation, is that when you feed into the same pathway, you oftentimes more than double the signal. It's a tremendous amplification. Exactly what you want in the gastric phase, because now you've got food in the stomach, so let's go full bore for acid production. The other one is histamine. Histamine has its own separate receptor. This is going to work through an adenylate cyclase activation, making cyclic AMP, activating protein kinase A, and then protein kinase A will phosphorylate downstream proteins that will feed into this signal, the same signal as calcium, to increase the fusion of these vesicles with the plasma membrane. So now we have synergy between the acetylcholine binding to its receptor, gastrin binding to its receptor, ramps up our signal, and now we're going to add a third histamine that is going to use a separate pathway to even feed more into that hose. So once we have these two hormones in conjunction with the vagal stimulation, then we have a full gastric phase response, and we have full acid secretion potential. So basically what we've done is like on a radio. Cephalic phase, turn the volume up just a little so you can hear that radio. When we get into the full gastric phase, now we're going to start turning that volume way up. So now, let's talk about gastrin and histamine. Where do they come from and how are they regulated? Before I talk about that, we have to talk about a general principle in GI hormonal control. And this is the endocrine system in GI. So there's two basic principles for the endocrine cells in the GI tract. One is called an open endocrine cell. The other one is called a closed endocrine cell. And all this simply means is where the physical location of the cell is in relation to the gastric lumen. So if we have an open endocrine cell, we will have a cell that looks like this. They tend to be a flask-like shape, where a small amount of the surface of this cell will be adjacent to other cells, and it will be incontinuous with the lumen, the gastric lumen. So it's open to the lumen. Now what this allows this cell to do is to take signals in. It can sense, it's actually a sensor, that it can sense what's going on in the gastric lumen. Sensing means binding something, because remember, the cell has the plasma membrane, so it has to communicate with its outside environment. It's only going to do that through receptors. So this cell has receptors that can identify material in the lumen of the stomach. Once it identifies this material, it can send a signal through the cell and cause fusion of vesicles with the basal surface of the cell, and now we can put the hormone into the circulation. So you can already imagine what a closed system is. If this is the lumen of the stomach, these cells are located in the submucosa, well deep into those gastric pits, but nowhere near in contact with the gastric lumen. The other name for these is enteroendocrine cell, abbreviated ECL. Entero meaning inside, endocrine cell. So in order for these cells to function, they have to receive some sort of hormonal signal, either in their local environment, paracrine signaling, or a signal coming from the blood, which is genuine hormonal signal. So this is coming from a paracrine or an endocrine signal. Now, with this information, we can define exactly how we obtain the gastrin and the histamine. So we have a cell, and I think you recall probably Dr. McGuire telling you that the endocrine cells of the GI system are named by the alphabet. Not very creative. So this particular cell is an open endocrine cell, and it's called a G cell. Now the easy way to remember that is, guess what hormone it makes? Gastrin, yeah. So this is going to make gastrin for us. It's an open endocrine cell, so it's open to the lumen of the stomach. And now in the stomach, what we have is digestion going on at this point. And we have receptors on this portion that are able to bind peptides plus amino acids. So this is our sensing function by the G cell. Once it binds these peptides and amino acids, it sends a signal to these vesicles to fuse, and out comes gastrin. Now the gastrin is going to have two functions. The first thing it's going to do is what we've seen. It's going to bind to the parietal cell in order to activate the signal transduction, to fuse those vesicles with the surface to allow the expression of those potassium proton transporters to get the acid production going. It's also going to bind to an ECL cell. The ECL cell, once it receives the gastrin binding, is going to secrete histamine. And of course, then the histamine bind to the parietal cell. Bless you. Now that's the hormone. We talked about the parasympathetic activating the parietal cell, and the parasympathetic will also send out acetylcholine to bind to the G cell, as well as the ECL cell. And this will also activate gastrin and histamine secretion. So what we have is a cephalic phase that's all the vagus nerve, and that's going to get some of the acid pumping transporters out onto the surface of the parietal cell. It's also going to stimulate the G cell to begin secreting gastrin, but not at a full throttle rate. It'll also activate the ECL cell to start secreting histamine to bind to the parietal cell. So that's the cephalic phase. Again, priming the pump, just getting everything ready. Then, once we have food in our stomach, we want full throttle. That's when the G cell really becomes activated, secretes a lot of gastrin, activating histamine secretion. Now we have another sensor that binds to the G cell. And these mechanoreceptors, of course, are located on the gastric wall. Mechanoreceptors are simply going to be measuring our extent of our Thanksgiving dinner. As that stomach starts to expand and starts to stretch, there's stretch receptors, that will send an impulse to the G cell. Now the neurotransmitter is very unique on this particular nerve, and it's called GRP. And GRP equals gastrin releasing peptide. So it's by all of these stimulations, sequentially stepped up in intensity, depending if you're in the cephalic or the gastric phases, that are going to really either get the pump started or go on to full activation. Now, how do we turn this off? Because once we have this activated, we have to find a way to turn the pH increases down, turn that pump off, retract those pump from the surface. We have another open endocrine type cell. This is called a D cell. Now the D cell, because it's an open endocrine cell, it has communication with the lumen, and this is going to be recognizing the decrease in pH. So it's a proton sensor. We have the D cell now putting out somatostatin that can actually put a brake on these two cells. Okay? Once we decrease the concentration of gastrin and histamine, we no longer have that positive signal going to the parietal cell. And so retraction of those transporters can occur. Once the transporters are retracted, then the proton pumping will end. Now, a principle of potentiation and synergy. This is really important when Dr. Shuttleworth talks about the GI pharmacology of acid control. So if we look at if we measure proton secretion and we measure a concentration of gastrin, would see is normal kinetics that we would expect. That looks something like this. It's a sigmoidal effect. So that the higher the concentration of gastrin we're adding, we finally get to a threshold activating the gastrin receptor, and now we start seeing proton secretion by the parietal cells. Eventually it saturates out. If we ever got that far, we don't get that far physiologically, but we would see that we'd saturate the receptor in signaling, and of course it would dampen out there. Now, this is gastrin. If we add histamine on top of this, because we see the signal with the parietal cells, a combined effort between the gastrin and the histamine, what we're going to do is we are going to now shift this curve to the left. So now, we will see the effect like this. So if this is gastrin alone, and this is gastrin plus histamine, we see now that a lower concentration of gastrin, when combined with histamine, will actually give a better effect. And this is the process called potentiation. Salivary secretions. Alpha amylase. Amylase is one of the first digestive enzymes that your food's going to see that's going to break down the carbohydrate, starch, into glucose. Now in the stomach, we're going to start beginning our protein digestion. We're going to talk now about chief cells. Chief cells are also located in those gastric pits, and they're going to be secreting what is called a zymogen. And the name of this zymogen specifically is called pepsinogen. So a zymogen is a general name for proteases that have no protein degradation function yet. Pepsinogen cannot degrade proteins. It's synthesized and secreted into the gastric lumen as an inactive protease. What we need to do is activate this to pepsin. The pepsin now can take your proteins and turn these into peptides plus amino acids. The activation of this you want under controlled circumstances. Because remember, what's lining your gastric lumen? Cells. Cells with proteins on them that have normal function. You don't want to start what's called autodigestion. Autodigestion means that you're secreting an active protease that's going to do harm and damage rather than target. So the cells, chief cells, secrete this as a zymogen, pepsinogen. The acid environment, the low pH environment, because of the HCl secretion, activates this to pepsin. Beautiful timing, isn't it? So as soon as food gets into your stomach, you increase acid production, boom, you activate your pepsinogen, and now you can begin your protein digestion. What do these do, peptides and amino acids? Gastric phase. They activate what? What type of endocrine cell? G cell, right? This is the activators of the G cell. Perfect again. You get your acid secretion going, you begin your protein digestion, now you generate the substrate to activate gastrin secretion to put out more HCl. That's all part of the gastric phase. Because again, once you get food into your stomach, pedal to the metal. You want full production of everything in that stomach lumen. Mucus. So what else does mucus do?
Protects from acid.
It protects from acid. And so along the lining, the gastric lining, all of these epithelial cells, the majority of them can secrete mucus. Now, this mucus is not going to be leaving the surface of the cell, because remember, in the salivary secretions, we talked about mucus, what it is. These mucin-like proteins, they've got short little protein backbones, they've got lots of carbohydrates to make them very water retentive and sticky. So they're going to line the lumen. It's going to be covering that lumen to provide a physical barrier. These cells that do this are called goblet cells. And I got a picture of one there in the notes. And it's called that because it looks like a goblet. And they tend to be very transparent looking because of all the mucin in them. Dr. Hansen, when he talks about the pathology of GERD and Barrett's, he'll be bringing in some much better pictures, and you'll see that the entire lumen is just lined with these cells. So they produce mucus, these mucin proteins, for protection, but they're also secreting something else that's very, 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 very critical. They're secreting bicarbonate ion, and this, plus the mucus, the mucin proteins, are going to line the epithelia. And it's these two factors that are providing the major protective function for the gastric lining. So you know the mucin is protecting against physical injury because lots of mechanical activity going on from peristalsis. Why do you want bicarb in there? Neutralizes the acid. Now, here's the real cool design of this. Mucus is very thick. It doesn't have a great diffusion rate. So the bicarb is secreted along with the mucin, and so very little bicarb ends up in the lumen. So you don't want to dampen out your pH change, because that pH change is activating your pepsinogen to pepsin. What else is it doing to proteins? Denaturing your proteins. Remember, acid conditions denature your proteins. That's going to make your proteins even more susceptible to pepsin activity. So this bicarb that these cells are secreting stays right into the luminal layer, right into that mucus layer. So you've got a pH of approximately 2 in the lumen. What do you think the pH is of the lining of the gut? Greater than that. Take a guess. What is a pH that is stable for cellular proteins? Is that low? I'm impressed. Okay, I'll be calling on you for the biochem. Poor decision. Poor decision. That's right, he's bigger than me. You got to judge carefully who you're going to call on, right? It's going to be about pH 7, about neutrality. And so that bicarb performs a very, very important function to protect. So now, think about a patient that has an acid problem. How does an ulcer come about? What is the acid doing? Eating. More specifically than eating. It's not really phagocytizing the cell or anything like that. So the acid is causing injury. It gets into the cell, it's damaging the apical surface of the cell, damaging the goblet cell, the mucus-producing cells. Then what do you have? You have an exposed surface that can be exposed to the acid and mechanical damage. Right? No mucus production because you damaged your cells. No bicarb production because you damaged your cells. And now you've got constant erosion of that epithelial layer because of that chronic acid exposure. Okay? So how do you solve an ulcer? There's actually a combined therapy. And Dr. Shuttleworth is going to tell you all about it. But one of the things, as Dr. Lynn would say, you know this. If acid is causing all the problems, Neutralize the acid, right, exactly. And then he'll talk about GI pharmacology on the methods on how to control it. There's also this thing called H. pylori. We'll get into that. Yeah. Okay, so the last secretion is intrinsic factor. Let's talk about that because I'm running over. Let's talk about that when we get, in about 3 weeks, when we get to vitamins, when we get to the B vitamins, and I'll go over that in detail.